Hello viewers, welcome to our video. We are so happy you're here. My name is Imelda. I'm a senior at St. Mary's College.
Hi, my name is Connor, and I'm a student at St. Mary's College. I got a question for you, Imelda.
What's your question, Connor?
Have you heard of an ELISA?
I sure have, Connor. ELISA stands for Enzyme-Linked Immunosorbent Assay. ELISAs use antibodies to detect antigens in a patient sample. Many pathogens can be detected or diagnosed using an ELISA.
That's cool. I bet you could look for antigen related to pathogens such as the flu, HIV, SARS, the Zika virus, and a whole bunch of others. But we're not here to talk about those viruses, are we?
I don't think so.
Well, today we're going to be talking about SARS-CoV-2, or as it's more commonly known, the coronavirus.
But first, let's quickly go over the basics of the immune response. Foreign pathogens will produce antigens, and our immune system will detect and respond to these antigens by producing antibodies. The antibodies bond to the pathogens, marking them for destruction by immune cells.
Antibodies have two regions, the variable region and the constant region. The variable region, located at the ends of the Y-shape, are highly specific and will bond to antigen, effectively marking them. The constant region, the stem of the Y-shape, communicates with the receptors of immune cells, telling the immune cells to destroy the antigen.
Let's talk about the infection timeline. After infecting a person, a virus multiplies in the body and antigen concentration increases. After infection, the immune system starts producing antibodies. As the antibody concentration goes up, these antibodies bind to the antigens, flagging them down for destruction. Eventually, the viral antigen load decreases and the infection is cleared.
Well, now that that's out of the way, let's talk about how we can use ELISA assays to detect the presence of SARS-CoV-2 in patient samples. First things first, we take a sample from someone who is being tested for COVID-19, perhaps someone who is symptomatic. We then add the sample into a well in a plastic microstrip plate with 12 wells so we can run multiple tests for each sample. We add the sample in and after 5 minutes remove the fluid. The sample will bind to the plastic in the well, so by removing the fluid, we're not removing the sample.
We then add wash buffer to the sample in order to rinse out any proteins that are not bound to the walls. This prevents anything from binding non-specifically in future steps. Next, we add the primary antibodies to the well. These antibodies bind only to specific antigen in the well. In our case, the antigen is the S protein.
The well is then incubated for 5 minutes and the fluid is removed. Buffer is then added to the well to rinse anything that is not bound. If antibody is not bound to antigen, it is washed out in this step. The wash step is then repeated once more. Bye-bye, unbound antibody.
Enzyme-linked secondary antibodies are then added to the well. These antibodies bind to the primary antibodies that are present. The secondary antibodies are covalently linked to the enzyme.
The sample is once again washed with the buffer to remove any secondary antibodies that are not bonded to the antigen. After this, we add a substrate that will react with the enzyme bonded to the secondary antibodies, producing a new color when activated. We can tell if someone is COVID positive if their ELISA changes colors, indicating the presence of the viral pathogens. If there is no color change, then that means there are no pathogens. But what about people who tested positive and want to see if they have any antibodies?
In order to see if a patient who tested positive for SARS-CoV-2 is making antibodies as an immune response to this pathogen, we would take a sample from the COVID-positive patient and repeat the steps of the ELISA, skipping the primary antibodies and instead adding the secondary enzyme-linked antibodies to the sample. These secondary antibodies would bind to the primary antibodies and would change color if exposed to the substrate. If there's a color change, then this indicates that the presence of anti-COVID antibodies are produced by the patient.
If there is no color change, then that means that there were no primary anti-COVID antibodies for the secondary enzyme-linked antibodies to attach to. Alright, now it's time for some additional questions. First up, in an antibody detection ELISA, lab-purified antigens, such as a coronavirus spike glycoprotein, or S protein, is added to the wells first. How is this step different than the first step in an antigen detection ELISA? Well, this step is different because we no longer need to test for the presence of the antigen. Instead of adding a sample that may or may not have an antigen in it, we know full well that the purified antigen sample has the S proteins for us to test for. So after the antigen is added, the wells are washed. Why is this step important? Give 2 reasons. The washing step is important for many reasons, 2 of which are that it removes any nonspecific proteins that may interfere with the ELISA, and also it helps to purify the sample added by making it so that the antigen is present in a higher proportion concentration within the well. If the sample in a COVID detection ELISA turns blue, a positive result, the patient sample contains antibodies to coronavirus. Does this necessarily indicate an ongoing infection? Explain your answer. This does not necessarily mean that the person is still afflicted by COVID, as there is still the possibility of the antigen being held onto after the infection to help the lymphocytes continue to produce antibodies to ward off any future infections. So, here we can see a graph showing the concentration of antigen and antibody over time in a patient. This graph is provided to us by Bio-Rad Laboratories. Thank you, Bio-Rad. Could an antigen detection ELISA be used at day 35? And what day would using PCR to detect the presence of viral genetic material be more effective? So at day 35, the antigen concentrations are likely too low to get an accurate result from an antigen detection ELISA. To detect viral genetic materials with PCR, day 15 would yield good results as the antigen concentration is highest then, signifying the peak of infection. At this point, it would be most likely that we would be able to replicate the viral genetic material using PCR. In the antigen detection ELISA, scientists often use antibodies produced by rabbits to bind to the antigen as primary antibodies, and antibodies produced by goats that bind to the rabbit proteins as the secondary antibodies. Could goat anti-rabbit secondary antibodies be used to detect 2 different types of rabbit antibodies? If HIV antigen was injected into the rabbit instead of influenza, could the same secondary antibodies, goat anti-rabbit, be used to detect both rabbit anti-influenza antibodies and rabbit anti-HIV antibodies? This image is brought to you courtesy of Bio-Rad Laboratories once again. Thank you, Bio-Rad. So the anti-rabbit secondary antibodies could be used to detect 2 different types of rabbit primary antibodies, as the anti-rabbit antibody targets a constant region of the rabbit antibody, which is largely similar across different types of antibodies. Depending on the similarities between the constant regions of the rabbit-derived anti-flu and anti-HIV antibodies, the goat anti-rabbit antibodies could possibly use to detect both antibodies from the rabbit. The more similar the constant regions, the more likely the goat antibody could detect the rabbit antibodies. Now, what would happen if the enzyme was linked to the primary antibody instead of the secondary antibody? Would the color still change upon adding the substrate? How would the intensity of color change be different? What are the advantages of using a secondary enzyme-linked antibody over a primary linked? The color would still change as long as the enzyme and substrate are still compatible with each other. This would produce the same reaction and therefore the same color change. Now the color change would likely be weaker than in ELISA using a secondary enzyme-linked antibody because multiple secondary antibodies can link to one primary antibody, meaning that there's generally a higher concentration of secondary antibodies. And one advantage of the secondary enzyme-linked antibody is that the higher concentration makes it so that it would be easier to tell if the color changes, easier to tell a positive ELISA result.
Thanks for watching and have a nice day. Please don't forget to smash that like button.